Tissue culture. Tissue culture relies on the fact that many plant as well as animal cells have the property of totipotency, whereby they can regenerate a whole organism just from a single cell of the body. Tissue culture is a practice used to propagate plants or animals from a small piece of the tissue excised from the plant or animals in a suitable growth medium. The steps involved in tissue culture should be strictly carried out under aseptic or sterile conditions, possibly in a sterile cabinet. A small tissue or a cell, known as explant, is taken from the growing part of the plant and placed in a culture medium. The culture medium contains a jelly-like substance known as agar, along with the proper mixture of nutrients such as sugar, vitamins and hormones. It helps the plant tissue to multiply quickly. Plant tissue divides rapidly to produce a shapeless lump of cell known as callus. Callus is transferred to another nutrient medium containing plant hormones, auxin and cytokinin. These hormones stimulate the development of roots and shoots in the callus. The callus, having roots and shoots, separates into tiny plantlets. These plantlets are then transferred to the soil where they can grow to form a mature plant. Plant tissue culture offers several advantages over traditional methods of propagation. It is possible to produce exact copies of plants that produce particularly good flowers, fruits, or have other desirable traits. The production of plants under sterile conditions reduces the chances of transmitting diseases, pests, and pathogens. It has become possible to produce disease-resistant varieties of plants, especially ornamental plants. Very little space is needed for developing new plants by tissue culture. Spore formation. In a previous activity, we have observed that the fungus grows on a piece of moist bread and forms cottony mass. Let us study reproduction in fungus. The white cottony mass is formed by the growth of fungus rhizopus or bread mold. The hyphae of bread mold or rhizopus are thread-like structures. There are rhizoidal hyphae which grow in the substratum. The mold synthesizes spores inside a sporangium. Sporangium is present at the tip of the erect hyphae. When the spores are ready to leave, the sporangium breaks open. The spores are released outside. If the spores land in a moist place, they germinate and form new mold. This method is known as spore formation. The fungus rhizopus reproduces by spore formation. Let's do a quick check.
Plasmodium. Spirogyra. Fragmentation. Potato. Vegetative growth. Rhizopus. Hydra. Budding. Leishmania. Binary fission.
Sexual reproduction. Sexual reproduction is another mode of reproduction observed in living organisms. Two parents are involved in sexual reproduction. The sexes are separate in higher animals. One male and one female parent are involved in sexual reproduction. Mainly, two processes are involved in sexual reproduction: meiosis and fertilization. Meiosis is a process in which the number of chromosomes is reduced to half. The reproductive parent cell is diploid with two sets of chromosomes. During meiosis, reduction division occurs, due to which the number of chromosomes is reduced from 2n to n, resulting in the formation of haploid cells with a single set of chromosomes, that is, n. These haploid cells undergo mitosis or equational division and form two identical cells each. Hence, we get 4 haploid gametes from a single diploid reproductive parent cell.
हम वैसे भी बोलते हैं ना कि 23 pairs of core chromosomes, 23 pairs. तो ये जो पहले डिप्लॉइड होता है वो एक पेयर होता है, फिर वो ब्रेक होके हेप्लॉइड में कन्वर्ट होता है, तो वो जो क्रोमोसोम की पेयर थी वो सिंगल क्रोमोसोम में कन्वर्ट होती है, फिर उसमें से अगेन माइटोसिस मतलब कि एक सिंगल सेल में से फिर से डिवीजन होके 2 सेल बनते हैं. दैट मीन्स कि एक डिप्लॉइड में से हमें 4 सेल्स मिलेंगे. ठीक है, डिप्लॉइड पहले क्रोमोसोम की पेयर थी, वो पेयर ब्रेक होके दोनों अलग अलग क्रोमोसोम्स हो जाते हैं, फिर उसमें से अगेन सिंगल सेल में से, जिसे हम बोलेंगे हैप्लॉइड, सिंगल सेल में से 2 नए सेल बनेंगे. ठीक है, दोनों में ऐसा होता है, मेल में स्पर्म, फीमेल में ओवम.
Meiosis is followed by fertilization, in which the haploid male gamete fuses with a haploid female gamete. This fusion results in the formation of a diploid zygote. This process restores the number of chromosomes from n in the gametes to 2n in the zygote, similar to the original parent cell. In sexual reproduction, since the zygote is formed from cells inherited from 2 different parents, the offspring produced are somewhat different from each of the parents. Variations give rise to variety and diversity. They enable the organisms to adapt and survive in a constantly changing environment. Variations help to maintain the species and thus prevent the complete extinction of animal and plant species. Why the sexual mode of reproduction? The basic event in sexual reproduction is the creation of a DNA copy. Cells use chemical reactions to build copies of DNA. This involves copying of the DNA as well as the cellular machinery. During DNA copying, there is a possibility of errors, due to which the DNA copying mechanisms cannot be considered as absolutely accurate and reliable. The occurrence of errors during DNA copying mechanisms is the source of variation in an organism. Several of these variations can be useful for the survival of the organism. Hence, there is a need to involve reproductive modes that allow more and more variations to be generated. The generation of variations within an organism is a fairly slow process. It is essential that the process of making variants should be speeded up to introduce favorable variations in an organism. During DNA copying, new variations are added to the already existing variations that have been inherited from the previous generations. The pattern of variation differs in different individuals. Combining variations from 2 or more individuals would create novel combinations of variants. Such a process of recombination of DNA can occur only during sexual reproduction. During sexual reproduction, the DNA from the male and the female gametes combines together to form the DNA of the zygote. However, the amount of DNA in the zygote does not get doubled. This is because the reproductive cells are haploid in nature and contain only half the amount of DNA or half the number of chromosomes as compared to that observed in the normal body cells of the organism. So, when a male gamete fuses with a female gamete during sexual reproduction, the zygote formed receives the normal number of chromosomes. Now, if the zygote has to grow and develop into a new organism with highly specialized tissues and organs, it needs to have sufficient stores of energy. This means that either of the germ cells should be developed and act as a source of energy. In case of simple organisms, there is no remarkable difference in the germ cells. However, with the increase in the complexity of the organisms, the germ cells also specialize. One of the germ cells becomes larger and stores the food material. It is known as the female gamete. The other germ cell is smaller and likely to be motile. It is known as the male gamete. This variation in the type of germ cells has given rise to differences in the male and female reproductive organs, leading to differences in the bodies of male and female organisms respectively. Let's do a quick check.
Meiosis is a process in which the number of chromosomes is reduced into half. False. Eman try na beta. શું થાય છે meiosis-માં? જે 23 pairs હતી એ સેપરેટ થઈને સિંગલ થઈ જાય છે કે નહીં? બરાબર? અને પછી માઇટોસિસમાં ફરી એ સિંગલમાંથી બે સેલ આવે છે. આવે છે ને? The basic event in sexual reproduction is the creation of a DNA copy. Process of reproduction does not allow more variation to be generated. False. The occurrence of errors during DNA copying mechanism is the source of variation in an organization. The DNA from the male and the zygote gametes combines together to form the DNA of the female. Only female DNA of the offspring. Zygote માંથી offspring.
Sexual reproduction in flowering plants. Activity. Visit a garden and observe a hibiscus flower plant. It is a shrub which bears solitary flowers. Pluck a single flower and observe its parts carefully. The flower has a long stalk on which all other floral parts are arranged. There is a receptacle at the end of the stalk on which sepals, petals, stamens and carpels are arranged. Sepals are united, forming a cup-shaped structure which is green in color. The petals are 5 in number and are brightly colored. This is the large showy part of the flower. Stamen is the male reproductive organ of the plant. The stamens are raised above the petals. It consists of anther and filament. The delicate filaments bear anthers at the tip, which are yellow in color. The filaments are united, forming a central tube, which starts from the base of the petals. We can observe a branch structure protruding from the central tube. This is a part of the carpel. Carpel is the female reproductive organ. It has a cap-like structure at its tip on which some of the yellow-colored pollen grains are attached. When we trace back this structure up to its base, we see a swollen structure at the base. It is the ovary which is present at the end of the long tubular structure. The flower is a reproductive organ of a plant. The different parts of a flower can be clearly seen if we take a longitudinal section of the flower. Flowers may be unisexual or bisexual. Unisexual flowers bear either stamens or carpels on a single flower. The flowers of papaya and watermelon are examples of plants bearing unisexual flowers. The flowers which bear both stamens and carpels in the same flower, such flowers are called bisexual flowers. Hibiscus and mustard are examples of plants bearing bisexual flowers. Now, we will study the process of sexual reproduction in case of flowering plants. Flower is a functional unit of sexual reproduction. The sepals, petals, stamens and carpels are the different parts of a flower. The carpel and the stamen are the parts of the flower essential for reproduction. Carpel is a female reproductive part of a flower. It is present in the center. It is made up of three parts, namely stigma, style and ovary. Stigma is terminal part of style, which is sticky in nature. It is a receptive organ onto which pollen grains will germinate. Style is an elongated part of carpel which bears stigma at its tip. Ovary is the swollen lower part of the carpel containing one or more ovules. Each ovule has an egg cell which is the female germ cell. Stamen is a male reproductive part of a flower. It is made up of two parts, namely anther and filament. Anther is usually bilobed and it produces pollen grains. Male germ cells are produced by pollen grains. The filament is the stalk of anther which raises the anther at the upper level. If carpels and stamens are necessary floral parts for sexual reproduction, then what is the function of sepals and petals in the flower? The sepals are usually green in color and they perform photosynthesis. They protect the floral parts in the bud condition. The petals are colored parts of a flower. The function of petals is to attract insects. Petals protect the reproductive organs which are situated on the inner side of the petals. Some of the flowers have attractive colors to attract insects for pollination. Pollen grains get stuck to the legs of insects. They are carried to the other flowers when the insect moves from one flower to another. These pollen grains germinate on stigma. Some plants have fragrance for attracting insects for the purpose of pollination. Let us now understand the process of pollination. Pollination. The process of transfer of pollen grains from anther to stigma of a flower is called pollination. When the transfer of pollen occurs in the same flower or on another flower of the same plant, the process is known as self-pollination. If the transfer of pollen occurs from the anther to stigma of the same flower, then the process is known as autogamy. If the transfer of pollen occurs from anther of one flower to stigma of another flower on the same plant, then the process is known as geitonogamy. If the pollen is transferred from anther of flower of one plant to the stigma of flower of another plant, then the process is known as cross-pollination. The agents of cross-pollination are wind, water or animals. The stigma acts as a receptive organ situated on the terminal part of style on which pollen germinates. Now, let us study about the process that takes place after the pollen lands on the stigma. Fertilization. The pollen transferred from the anther germinates after it lands on the stigma. The pollen tube grows out from a pollen grain. It travels through the style to reach the ovary. Each pollen tube contains 2 male gametes. The male gametes are released near the egg cell present in the ovule. One male gamete fuses with the egg cell to form a diploid zygote. The second male gamete fuses with a secondary nucleus in the embryo sac to form endosperm. Endosperm is a nutritive tissue for the growing embryo. This process is called double fertilization because the 2 male gametes fuse with 2 different cells in the ovary. The zygote develops into an embryo. The embryo is capable of growing into a new plant. We will now study how an embryo develops further into a seedling with the help of an activity. Activity. To understand the process of development of embryo into a seedling. Soak few seeds of Bengal gram in water and keep it overnight. Drain off the excess water and cover the seeds with wet cloth. Keep it undisturbed for a day. The next day, open a seed carefully and observe its different parts. We can see that the seeds absorb water and become turgid, which marks the beginning of the process of germination. The radicle comes out of the seed. Due to the pressure created, the outer seed coat is ruptured. On the same axis of radicle, on the upper side, we can observe the swollen part of plumule. The fleshy white cotyledons store the food material. The process of development of embryo into a seedling is called germination. Let us now study the process of germination. After fertilization, the zygote divides several times to form an embryo within the ovule. The ovule develops into a seed. The ovary develops into the fruit. The seed contains the future plant, that is, the embryo. The seeds show outer seed coat which breaks open so that the embryo further develops. Embryo shows radicle or the future root and plumule or the future shoot. The seed develops into a seedling under favorable conditions. This entire process is known as germination. Let's do a quick check.
कौन सा सही है?
Reproduction in human beings. We have already studied the different modes of reproduction in unicellular organisms and plants. Let us now study the mode of reproduction in human beings. In human beings, the reproductive system of males and females are different, with different organs performing a specific function. For example, special reproductive organs to produce sperms and eggs. Human beings can only reproduce after reaching sexual maturity. So, let us discuss about the age at which a person begins to mature sexually the changes that occur in the body after sexual maturation, and the causes of these changes. We all undergo many changes from the time we are born. Our height and weight increases. We acquire milk teeth and even lose them. We again acquire a permanent set of teeth. These all changes can be considered under the general process of growth. But there are a whole new set of changes during the early teenage. The appearance of the body changes, new features starts developing, and so do the new sensations. Some of these changes are common to both boys and girls. Growth of thick hair under armpits and in the genital areas can be noticed. The genital area, that is, the area between the thighs, grows darker. Development of thin hair can be seen on hands, legs, and even on face. The skin becomes oily and is prone to develop pimples. There are few set of changes that are not common in both boys and girls. In girls, breast size increases with darkening of the skin of the nipples at the tips of breast. Girls start menstruating at around this time. Boys develop thick hair growth on face and their voice becomes hoarse. All these changes take place over a period of months or even years. The changes do not happen all at the same time in one person, nor do they happen at an exact age. Some people acquire these changes too early, while in others they happen very slowly. Similarly, each change does not occur or become quickly. All these changes are the aspects of the body depicting sexual maturity. Do you know why does the body start developing these characters at a particular age? In multicellular organisms, specialized cells perform specific functions. Plants develop special cells and tissues to carry out the process of fertilization. Similarly, humans develop specialized tissues and germ cells to participate in sexual reproduction. After crossing the age of 10 or 11 years, there are sudden noticeable changes in the growth pattern. The body begins to undergo rapid changes, leading to reproductive maturity. The period of life when the body undergoes these changes is called adolescence. The age of adolescence varies greatly from person to person. It begins around the age of 11 and lasts up to 18 or 19 years of age. As this period covers the teens, adolescents are also called teenagers. Adolescence in girls begins 1 or 2 years earlier than the boys. Adolescence ends at adulthood. The changes that take place during adolescence mark the onset of puberty. The period of life when the body becomes capable of reproduction is known as puberty. It normally starts at 12 to 14 years in boys and 10 to 12 years in girls and varies from person to person. Several changes occur in the body during puberty. These changes do not occur instantaneously. It takes many years for the body to undergo these changes. Puberty ends when an adolescent reaches reproductive maturity. How are all these changes related to the process of reproduction in humans? The sexual reproduction means fusion of two germ cells from each individual to form a zygote. This can happen either by release of germ cells from the body of an individual externally, as in case of flowering plants, or it can happen by internal transfer of germ cells for the fusion, as in case of many animals. Humans can undergo the process of sexual reproduction only after attaining sexual maturity. Thus, the changes that take place during the puberty are the signals that sexual maturation is taking place. On the other hand, the actual transfer of germ cells or gametes between two individuals requires special organs for the act. For example,
ये organs के बारे में हम बाद में देखेंगे.